Hi, my name is AJ from We Learn to Share, and today we'll be learning
Unit 5 of the official AP coursework, Heredity. And I divided this chapter into 2 lectures, and this will be part 1, the cellular basis of heredity. So let's get started. Right, so here is the table of contents. Number 1, molecular basis, what are chromosomes? Cell cycle, what is the process of cell division? Number 3, mitosis, how do cells replicate and separate? Number 4, factors of cell division, what conditions do cells require to divide? Number 5, cell cycle control systems. How is each phase in cell division controlled exactly? And number 6, meiosis and genetic diversity. And what is the significance of meiosis and how does meiosis contribute to genetic diversity? Right, so what is reproduction? Reproduction is a production of an offspring, whether by asexual or sexual mechanism. The asexual mechanism would produce a clone, that is, another organism that has the exact same set of genes and chromosomes, and at least at the molecular level, designed to be same as the original organism. And for sexual reproduction, we are products of sexual reproduction to start with. We receive half of our genes from each of our fathers and the other half from each of our mothers. This mechanism of half and half, mix and match, sexual reproduction gives us similar yet distinct features from our parents. So if you think about it, if your parents are tall, you tend to be more tall. And sometimes you might have a bigger nose if your father has a bigger nose. Or, yeah, something like that. So, next up, eukaryotic chromosomes. The characteristics of eukaryotic chromosomes. Normally, DNA isn't in the form of an X-shaped chromosome, contrary to what you would expect. They are in the form of chromatin, unwound version of DNA and proteins. Think of chromatins as long lines of threads. And the proteins that hold the DNA together are called histones, histone proteins. And as the genes prepare to replicate and divide, these chromatins, they coil up into chromosomes to facilitate the process of replication and division. And as the chromosomes replicate from a single chromatid chromosome, it forms a structure of 2 sister chromatids. It doubled their DNA. And it forms a structure of 2 sister chromatids. The shape you probably are most familiar of. So the shape you expect, this shape, the most famous one, this is after the replication with 2 sister chromatids. This each line is called a chromatid. And the region that 2 sister chromatids overlap, this part, is called a centromere. So, about the chromosomes in our body. As you may or may not know, humans have 46 chromosomes in somatic or body cells. Humans are diploid organisms. We receive one haploid set of genes from our fathers and the other set from our mothers. The genes we receive from each of our parents would then form a pair. We call them homologous pairs. When we refer to a location in a single chromosome, for instance, to describe a distance between genes in a single chromosome, we may refer each location as locus. The plural version is a loci. Of the 46 chromosomes that we humans have, 44 are not related to sex. And these 44 chromosomes are called autosomes. So when someone talks about autosomal disorders, these conditions, are not sex-linked. They are probably genetically mutated chromosome conditions, but they mean that they are not sex-linked. And the remaining 2 chromosomes, so we had 44 autosomes out of the 46 chromosomes, so 2 remains, right? So the remaining 2 chromosomes are either XX or XY. each resulting in female and male respectively. So simply put, if someone inherits the Y chromosome from that person's father, he is male. So if you have a Y chromosome, you're a male. If you don't, you're a female. Okay? So how do cells divide? What mechanism? Exactly how? So in order for a cell to divide, there are prerequisites. The part where the cell divides is actually only a very small portion of time in the cell cycle. The preparation time is called interphase, and it takes about 90% of the time in the cell cycle. Within interphase, there is gap 1, which increases cell size, and S phase, which replicates DNA. The S here stands for synthesis. So you're synthesizing DNA and replicating DNA. And then after that, there's gap 2, which is the last stop before the actual process of division, the M phase. So you prepare for the division. And it's also often said that this process, gap 1 and gap 2, are misnomers, since these aren't just simple gaps. They are an important part within the interphase, and it's important for the cell to actually proceed into the M phase. So yeah, don't sleep on that. Don't sleep on gap phases. Then next is M phase, mitotic phase. This phase is basically mostly mitosis. I forgot to mention what mitosis is. So mitosis is a replication process that results in two daughter cells that have the same set of genes as the parent cell. And so basically this is the most common process that the cells go through within the replication process. And after that, within the M phase, there's cytokinesis. And cytokinesis is the process that separates the cytoplasm. So as mitosis almost ends, the cytokinesis has also got to, in order for the cytoplasm to separate, there needs to be cytokinesis. Alright, take a good look at the cell cycle diagram, and I advise you to pause this video to really understand how this process works. So yeah, pause if needed, take a good look. Alright, now we're going to talk about the detailed stages inside mitosis. So as you can see, there's 5 stages within mitosis, and it starts off with the prophase. And in the prophase, chromosomes form and the mitotic spindle begins to form. For those of you wondering what mitotic spindle is a structure of microtubule fiber and associated protein that serves to guide the separation of the chromosomes. In the prometaphase, sometimes this process, the prometaphase, is omitted in some of the textbooks. So sometimes it just goes prophase, metaphase, anaphase, telophase instead of prophase, prometaphase, and metaphase. But still, we're going to include this. And prometaphase, basically, the most important part is that the nuclear envelope breaks, and then microtubules start to reach the chromosomes. And in the metaphase, the mitotic spindle is fully formed. And most importantly, the chromosomes line up in the middle, and the kinetochores attach to the microtubules. So, for your understanding, metaphase starts with the M, right? Meta, it lines up in the middle. So think of M and then middle. So metaphase is the phase where the chromosomes line up in the middle. And the anaphase is when the sister chromatids are separated and the poles start to move away from each other. And telophase is basically, they say telophase is the opposite process of prophase. And telophase is where nuclear envelope forms again, and then mitotic spindle disappears again. And I made this acronym for you guys to remember. So in order to not leave out any of the processes here mentioned, we're gonna do a little acronym exercise. And you know, think about a dog, okay? There's a dog, and this dog has to pee, right? Where does dog pee? Well, normally there would be an area for a dog to pee, right? And normally that area would be some kind of a pee mat, right? This mat, this blue, search up pee mat on Google, and there's this mat, blue outline, and inside there's a white tissue-like mat that, that these dogs pee on. So yeah, these dogs pee on the pee mat, right? These dogs pee on the pee mat. So when someone talks about stages of mitosis, think of the dog peeing on a mat, and then it would be pee mat. So PPMAT, prophase, prometaphase, metaphase, anaphase, and telophase. So yeah, this may sound a little bit dumb, but this is an acronym that I use, so good luck with that. And right, again, pause if needed. These steps are important. Take a good look. Next up, factors of cell division. There are some factors that need to be met for cell division. First up is anchorage. Cells need some kind of a surface to hold onto. So in order for cells to divide, there needs to be some kind of anchorage in order to replicate. Next, these cells detect the density and regulate their division. That's why normal cells don't divide to a degree that piles up. On the contrary, though, cancer cells are known for their uncontrolled growth, and they do not care about density as a factor. Then next, since replication obviously is a hard work that requires a bunch of resources for the cell, essential nutrients or growth factors are also very much needed. These control systems are cyclins and CDKs. And CDK stand for cyclin-dependent kinases. So basically, these are just cyclins, and then the proteins, the phosphorylating proteins that these cyclins depend that are dependent on the cyclins. So they are positive regulators, and in this positive doesn't mean that it's good, as in like morally good or quality-wise good. It's just recall positive feedback loop. And then CDK cyclin complex needs to be phosphorylated in order to be activated to phosphorylate other things. And the definition of kinase is an enzyme that phosphorylates other enzymes. So obviously CDK is a kinase, is a one kind of kinase, and obviously it will serve to phosphorylate other proteins to send out a signal and possibly amplify it. So the concentration of cyclins, different kinds of cyclins, are different for each distinct phases, and it's important to note that. Meiosis and genetic diversity. Meiosis, this is the most crucial mechanisms for genetic diversity. Important terms, meiosis is a crucial process in the sexual reproduction. Cells containing one set of chromosomes are called haploids. Cells containing two sets of chromosomes are called diploids. Meiosis is the formation of haploid cells from diploid cells. So before it was mitosis, correct? So this cellular division process of mitosis, it was formation of diploid cells, two diploid cells from a single diploid cell. But this different process, meiosis, is the formation of four haploid cells from a single diploid cell. And we'll review this obviously, but basically there are two stages of meiosis. Unlike mitosis that goes through a single process, there's two stages, meiosis 1 and meiosis 2. Not two different, it's not two homologous chromosomes, it's a single chromosome, and then the chromatids, it's the chromatids that are pulled apart instead of the whole chromosome. And in telophase 2, two haploid cells separate into four haploid cells, And then meiosis 2 is often said to be analogous to mitosis. So basically, since in mitosis, the two sister chromatids are separated from a single chromosome. So basically, meiosis is the same thing, but then in a different kind of a process. That was alteration of chromosome number, and this time is alteration of chromosome structure. And there are 4 main types of alteration of chromosome structure. And the first one is deletion, and which is a segment of chromosome being removed. And the next is duplication, segment of chromosome being copied. And inversion, segment of chromosome initially removed and reattached to the same chromosome, just in a different kind of a alignment. And the last one is translocation, which is a segment of 2 chromosomes swapping. So inversion, it's important to distinguish inversion and translocation. Inversion is within the same chromosome, and translocation is swapping between chromosomes. Right, so that's it for today's lesson. If you have a question, leave a comment down below. And next week, we will continue and finish our Unit 5 Heredity.